Welcome to another video tutorial. In this video, I will explain how we can interpret the results of metabolomic data and will also show you which figures are important for research article point of view. We will also talk about selection criteria of differential metabolites and recent tools being utilized in metabolomic study. So as we know that in metabolomics, first of all, we take the metabolites from our sources. It can be our plant samples or animal samples, and as we know that we usually used GCMS and sometime LCMS, and similarly NMR is also utilized to get different metabolites. And then we perform data analysis, and as we know that data analysis includes statistical analysis and bioinformatic analysis. So after this we also perform biomarker identification, and sometime we also do further integration of our metabolome with proteome and transcriptome and genome. So let's go into further detail that how we can start with metabolomic data. So to understand this, let's first talk about workflow of metabolomics. So as we know that, first of all, it begins with the compound detections, and compound detection can be done through mass spectrometry or through NMR. After we have detected the compounds, so we take them for data pre-processing step. And as we know that at data pre-processing step, we run some QC. QC stand for quality control. I mean, we go for noise reduction, we go for retention time correction. As we know, we usually get the data of metabolomics through GC or LC. So we get this data in the form of peak or mass to charge ratio. So there are some outliers, so we need to reduce them. And after we have done the peak detection, we also integrate and do alignment of chromatogram. So all these steps are performed by usually analyst, and after that we do data processing. And as we know that sometime our data is not normally distributed. I mean, sometime it is skewed to right or sometime it is skewed to left. So it is important to do data normalization, and we also perform compound identification. So although there are two ways of metabolites detection, I mean, maybe it can be targeted and sometime it can be untargeted. And as we know that in untargeted, we are not focused on any specific one. I mean, we just go with the journal profiling, and in the targeted one we go for the finding with the specific one. So I mean, in where we need to identify the targeted one, so we need to perform this step earlier. But if you don't want to go for any targeted metabolites, then maybe you can do this step later on at the third step. And if we can see, the next one is the statistical analysis. And usually the statistical analysis covers almost 95% of the analysis. I mean, in any metabolomic data, the statistical analysis covers up to 95% or 90%. And as we know that statistical analysis in metabolomics data is covered through univariate analysis and multivariate analysis. And as we know, in univariate analysis, parametric or non-parametric testing we can apply it. I mean, if the data is normal, so we go for parametric, and if the data is in the form of like ratios, so then we go for non-parametric testing along with simple t-test and other test. And we also do some fold change analysis. And as we know, in multivariate analysis, we perform multivariate regression analysis, and then sometime do ANOVA, PCA, and PLS, that is partially scared discriminate analysis. So we also call this PLSDA, partial least scared discriminate analysis. And then we have the functional analysis. I mean, almost this is covering almost 5% of all recent reported studies. So in this, we perform gene ontology and enrichment analysis and pathway analysis, which is done through the KEGG. And similarly, we also perform omics data integration with other like transcriptomes, proteomes, and microbiome, just to validate the results. So here was the journal flow of metabolomics, and now we will talk about what are those statistical analyses which are commonly used in metabolomics. So as I previously mentioned in slide, that we usually use univariate analysis and multivariate analysis. So what are those differences? So as we know that uni, as the name is representing, so if we have the single variable, so I mean at a time, so I mean we only need to compare one variable with one variable, so then we will use, I mean if you have only treatments like if you have wild type and like mutant type, if you now you see we have only one and one variable, so if you need to compare only single variable with another control or anything, so then you will use univariate analysis. And as we know, this is the simple one, and we compare one variable at a time. I mean we do comparison of one variable with one variable. And in the multivariate analysis, we can do comparison with like two or more variables at a time. And as we know, the univariate was a simpler, and the multivariate analysis is complex, and we can also perform two or more variables at a time. So sometime if you will send your sample to the company for metabolomic analysis, so they will perform univariate analysis and multivariate analysis together. I mean they will use univariate analysis to just give you comparison of one to one, and if you want to do multiple comparison, they might also perform ANOVA to go for multiple groups comparison. So we can talk about which folders are important in metabolomics analysis. Usually whenever you send your sample to the company, so the company send you the data is in bulk. So sometimes student ask me, I mean what are those folders, I mean they are confused which data is important. So let me show you in one example. So as here you can see, this was the result sent to me by a company. So as here you can see, here we have basic statistics, PCA and PLSA, that is partial least square discriminant analysis. I mean you can say this was all about the quality control, and this was the PCA, and we know that PCA is used for dimensionality reduction. I mean if you don't know in detail what is PCA analysis, so I will recommend you, I have uploaded another video which is covering on the interpretation of PCA, so you can watch that, and you will have a better idea that what is PCA. And similarly like PLSA and OPLSDA, so you can see this is actually the same dimensionality reduction. I mean whether the groups are falling within the same group or not. So but this is like the advanced one, so their purpose is actually the same. So we will cover almost I mean we will take only one figure from these three folder. And as in my metabolomic analysis, I have only one variable. I mean I was comparing mutant type with the wild type, so they have done the univariate analysis for me. And then I have potential biomarker analysis and pathway analysis. So all the important differential DEM. We call it differentially expressed metabolites. I will also talk about later one. So all the DEM file you need to dig out this folder, I mean univariate analysis for this one. So you can take most of the figure from this folder, and after that potential biomarkers, I mean, and then pathway analysis, we will also be choosing at least one figure from each as well. So as you know, in the previous videos, we have already discussed about I mean what selection criteria we used in normally like the proteomics or transcriptomics. We also use in proteomics, and as you know, in proteomics we used our DEP, differentially expressed protein. And as you know that we select the differentially expressed protein based on two things. One was FC value, and I mean if the FC value of differentially expressed protein was equal to more than 2, and the p value is less than 0.05, so then it would be your significantly differentially expressed protein. So similarly, here is the same selection criteria, but it may varies according to your need. But I mean, we usually use DEM. I can change the color. DEM we use for more than 2. So we do the selection of differentially expressed metabolites similarly. I mean, their FC value is more than 2, and sometimes we also do if the FC value is more than 1 also, but over 2 is recommended. And similarly, we can do with p value, and or maybe we can also do with FTR. And as we have already done in our transcriptomic or proteomic video, so their level was less than 0.05. So if you haven't watched those previous videos, with in that video I have discussed in detail that how we can like make a comparison of fold changes, and even how we can calculate up and down regulated differentially expressed metabolites, protein or transcripts. So you can go there and watch those videos first, and now we can move ahead. And as we know, usually we get our data in the form of figures. So here are the some graphical visualization which we get through two different softwares. So here the most commonly used software, although this is free, I mean most of the modules in this is free, but if you want to buy space, so you can also buy space in this MetaboAnalyst and Metflow. So I will talk about the figures generated through Metflow and MetaboAnalyst. So in this figure, all the figures are generated through this Metflow software. So as here you can see, the first figure is actually representing the RSD, that is relative standard deviation. So as here you can see in LCMS and GCMS data, in LC and GCMS, so we use 20% selection criteria for LCMS and use 30% for GCMS. And as here you can see, this is the selection criteria to select, and here are the peaks, and here is relative standard deviation percentage for metabolites. And here is actually the volcano plot. So as you know, this is showing the fold change value. I mean here you can see this was the 1 and this was 2, so it means here are those metabolites which are being increased. I mean the green color is showing the increase, and the red one is showing those metabolites which were decreased in your treatment compared to the control. And this thing is actually the heat map, and as we know that heat map is a good way to represent like the different group. As here you can see, in the blue one is being mentioned for the control, and the red one is actually mentioning the case. And as here you can see, here each line or each dot is representing one metabolite, one metabolite. As in the previous transcriptomic video, as I said that each line is representing one transcript. So here one line will be representing one metabolite. So here you can see this is the being upregulated and this is being downregulated, and similarly these are being downregulated as you can see through this key, and here they are being upregulated. So similarly heat map will be used as we have used in transcriptomic studies. And here is the PC analysis, principal component analysis score plot. And here is, you can see the case versus control. So here for the case and here are the control. So here you can see the PC1 is covering this much and PC2 is covering this much. So if you want to go for more details, so I will recommend you can first watch about piece what actually we can interpret from principal component analysis. So next, that is PLS score plot. So the purpose of PCA and PLS score plot is actually the same, but as here you can see, this is PLS score plot is more precise. I mean, now you can see they are more together in the case and control, and we can see a better look of groups and treatments. And similarly this is also OPLS score plot. So all these graphic visualization you can select from your folder for your own analysis. So these were actually generated through Metflow, and this software is also free. So as like the MetaboAnalyst, so you can create a free login for this website, and you can upload your on metabolites list, and you can do these all analysis by yourselves. So now I can also show you some graphical visualization generated through MetaboAnalyst on next page. So here are the actually graphical visualization generated through MetaboAnalyst. So as here you can see the same one. Here we are representing log FC through volcano plot, and here we have p-value. So as here you can see, this is the threshold for up, and I mean these are the upregulated metabolites and these are the downregulated metabolite. So you can see in this volcano plot, we can see we have more upregulated differentially expressed metabolites as compared to down-regulated expressed metabolites. So here is actually the score plot. So we can see we have PC1 and PC2 for our different sample. Like you can see here we have three groups. This green one was taken from mouse liver 2, and this blue one was taken from mouse liver 3, and this red one is taken from mouse liver. So there are actually clear differences. And again, as in the Metflow, we have heat map. As previously we have explained that we can have a comparison. As here you can see we have data wild type and knockout. So here we have their name, and here we have list of samples. And of course after statistical analysis we have enrichment analysis as we do in transcriptomic analysis. And here we have demo plot for, I mean, if your data for glycine, serine, threonine and metabolism was the, I mean, the topmost pathway, so they will also provide you the demo pathway. So I mean in the MetaboAnalyst it is a plus that you can also get the demo pathways. And here are also the demo pathway for glycolysis and gluconeogenesis. So here this rectangular box is actually representing gene, and this round is actually representing one metabolite. So one thing important to mention, I mean, if I can zoom in, I will later on zoom in the slides. Here you can see here are the different IDs. So I mean these IDs here you cannot search. If you want to search them, what actually these IDs are representing. So these IDs can be searched in the KEGG data base. I mean this ID is coding for metabolites. So if you want to get more detail about this enrichment pathway, so I have already recorded separate videos, so you can go and watch them to have a more clear idea. And now let me show you some detailed diagram generated through MetaboAnalyst. So when you will upload your data, so you will also get your ANOVA, I mean statistical analysis, PSA, PLSA, and all this. And you can also, when you will click on this, so you will see the graph. I mean, if you have used 4 level, you can see here. If you have 4 level, so you will have a comparison of each graph. So I mean, as here you can see in the 45%, the uracil concentration of the metabolite actually, uracil metabolite concentration was highest in the 45. And similarly we can also have a comparison of pattern matching. So as here you can see, let me change the color. So as here you can see, now we have actually the correlation of top 25 compound. So as here you can see, it is showing the plus with, I mean, if endotoxin is high, so it is giving you the high glucose. I mean this is actually showing the correlation of compounds with your actual detecting compound. I mean, if endotoxin is high, so it will increase the metabolites, glucose and alanine, methylalanine. I mean, the highest correlation was found in this, and similarly, the negative correlation was found in 3-PP and isobutyrate. So, I mean, this is good if we can have a correlation of different compounds and different metabolites. And we can also have comparison in the form of the enrichment score. As here you can see, the red the color, I mean the higher the p value. So in this, the most significantly enriched pathway was glycine, serine and threonine metabolism, and similarly protein biosynthesis. As the color will be red, so there are more chances that this is your candidate pathway, uh, controlling the phenotype of your, I mean whatever you are performing your analysis. So MetaboAnalyst will provide you a lot of opportunities to create these pathway visualization. And we can move ahead, and here you can see paint omics was used to create one interaction network. As here you can see, we have different metabolites and how they are playing their role in this anemia pathway. So here are the different colors. So the red one is actually being changed one. I can change the color. Okay. Yeah, the red one is actually being changed, and the blue one is actually just run as a control. So you can also have a better idea about your any specific pathway. So for the selection of any specific pathway, it all depends on, I mean, the literature. I mean, you can first identify your differentially expressed metabolites, and then you can see these differentially expressed metabolites, how they are related with your phenotype. I mean, which you have also performed any other analysis except this um, Metabolomics data. So then you can relate those differentially expressed metabolite with any pathway. Like if any metabolite was related to iron or anemia, so I mean, of course, then you can choose this pathway. But actually the selection of any pathway is very tricky. I mean, it's not only depending on the one factor, so there are many factors which may ultimately which may ultimately affect the selection of your final pathway. And here are also met pathways. So as here you can see, we have different pathways and negative log values. Here 3-omics was used. As you can see, now we have integrated, I mean, we not only getting metabolites, and we also get proteins and transcripts as well. So hopefully now you will have a better idea. I mean, what are those important graphs and figures uh, for the selection to make your own story for your research article, which can be generated through Metflow or MetaboAnalyst. But if you have still any question, please let me know in the comment section. And thank you for watching.